Hello. Going to continue here and talk about lymphocytes. This is chapter 22, the part 3. So T and B lymphocytes have unique receptor complexes. So they have their own kind of receptors here, and they are unique for the T cells and unique for the B cells, of course. Um, so lymphocytes come in contact with the antigen. And as we were talking before about antigens, this is something that causes an immune response. This was basically what causes the T and B cells to produce um, antibodies. This is what an antigen is. Now the T lymphocyte have different types. There's different types of them. They're helper, they're cytotoxic, and there are other types too. Memory cells, regulatory cells, T cells. So different types of T cells. The helper ones are called the CD4 plus, and the cytotoxic ones are called the CD8 plus cells. So helper T cells, CD4, help other cells activate, including themselves, and they help B cells. They start to initiate the entire um, immunological response that creates antibodies. Cytotoxic T lymphocytes are other kinds of cells that will release chemicals that will kill cells. So they fight the disease by killing cells. So T lymphocytes and B lymphocytes. Here you're looking at T lymphocytes, different receptors on there, the different types, T4, helper, T8, cytotoxic. And then over here is a B lymphocyte. So the whole process begins with what's called the antigen presentation, the antigen presenting cells, and major histocompatibility complex 1, 2, and 3. We're just going to talk about, we'll talk about 1 and 2 to start with here. So the antigen is presented by a specific type of cells. So antigen cells are phagocytic cells. Not any kind of phagocytic cell can do it. Dendritic cells can. Actually, B cells, B lymphocytes can be antigen presenting cells. Macrophages, what they do is they go and they go to where the infection is and they phagocytize whatever it is that's causing the bacteria or whatever it is, virus. They phagocytize it, and that breaks it up. Okay, they break it up once they get inside the cell, and then they take these pieces of it and they put it on this specific molecule called the MHC2. Okay, major histocompatibility complex. And you can see that on this one. Well, I won't get into that right now. Now there are different classes. So the MHC class 1, the major histocompatibility complex 1 molecules are glycoproteins. Every cell in the body has these, every single one of them. It identifies them as self to the immune system, but it also is a reporting mechanism to the immune system. And if they go making any, it presents proteins that it's busy making, just presents them. And so if those proteins are totally normal, hey, everything's fine, leave it alone, you're cool. If not, if it's a bacterial protein, if it's a viral protein, ding, ling, 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 alarms go off, and it's time to kill that cell to induce apoptosis, which is programmed cell death, and release noxious chemicals to help it die. Okay. So here you can see formation and docking of MHC class 1 molecules in a healthy cell. Okay, so there we go. Molecules continuously synthesized. Transport vesicles go through the rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi. They get presented on the surface. You can see here self-antigen, right? Yeah. Okay. So an unhealthy cell. So the MHC class 1 molecules are synthesized by the rough endoplasmic reticulum. Peptide fragments of the viral protein particle antigen become attached to the MHC. So this right here is a foreign antigen. As you can see here, this is not the same healthy protein that was made before, and it's presented on the outside of the cell. And when that's presented, um, MHC class 1 molecules with bound foreign antigen are displayed within the plasma following fusion circulatory vesicles. So remember, this one then is what we're talking about here is the cells are going to identify to the immune system that something is not right, something's unhealthy, something wrong is going on in this cell. Okay. So talking a little bit about the antigen presenting cells, the MHC class 2 molecules are also glycoproteins, another kind of class of those, but only a few cells have these, only cells that are antigen presenting, only those that are allowed to present an antigen to the immune system, basically. So display these on professional antigen presenting cells. Ooh, professional. Synthesize, modify the RAR exogenous antigens brought into the cell through endocytosis, phagosome release of fragments loaded on the MHC class 2 molecules. Okay, so formation and docking the MHC class 2 molecules in antigen presenting cells. So you can kind of see similar thing going on here. Here, let's explode this, expand this, blow it up, explode it. So here you can see that whatever is the antigen from the bacterium comes in, okay, comes into the cell, and it gets broken down into pieces, right, in the phagolysosome, combines with the transport vesicle and the secretory vesicles right here. The transport vesicle has got the MHC class 2 molecule in it, so it's being synthesized by the cell. So it has to go through the endoplasmic reticulum, the endomembrane system, remember that? And when it's done, it fuses with that phagolysosome. Okay, so phagosome, lysosome, and the secretory vesicle all come together. Transport vesicle come together in the secretory vesicle and gets displayed on the surface as a foreign antigen. All right, this is the alarms going off. Ding, ding, ding. And what it's saying here basically is that it's found this this potentially dangerous, potentially infectious particle, and it wants to present it to the immune cells, the cells of the immune system, for processing. Okay. So as we can see here, um, T cell receptors, this presenting an antigen, a foreign antigen, is a antigen presenting cell. Okay, presenting foreign antigen to the T cell receptors, like it says CD4 or CD8. Okay, CD8 will activate it to be a cytotoxic cell. CD4 will activate it to be a helper cell, but they're just as capable, causing the immune response, just in a different component of it. So, talking about the lymphocytes, we got their formation. So in the bone marrow, they differentiate and become all the way to matured B lymphocytes and pre T cells, pre T lymphocytes. And then those pre T cells will mature into active T cells in the thymus. Okay. Formation of lymphocytes again. Activation of lymphocytes. So we're talking about the formation of lymphocytes a minute ago. Now this is the activation of them. So once they've been activated, then they cause the immune response that we call the adaptive response. All right, so wait a minute, expand this. So the lymph nodes and spleens and tonsils. Okay, secondary lymphatic structures house B and T lymphocytes, site of activation, proliferation of these cells. So in other words, when it encounters it again, then it's ready. T lymphocytes, like we said, originate in the bone marrow, migrate to the thymus, pre-T lymphocytes to complete maturation. Um, selecting T cells, T lymphocytes. Thymic selection eliminates 98% of T cells produced. There's a positive selection, selects for the ability of T cells to bind thymic epithelial cells with MHC molecules. Those that can bind survive. All right, and negative selection tests ability T lymphocyte to not bind self-antigens, self-tolerance. Occurs in primary lymphatic structures, specifically called central tolerance. So what does that mean? Um, we are looking at T cells, and they are made. So T cells have, each one of them has a unique T cell receptor on it, each one. And so what happens is those T cell receptors will interact with the immune system, will interact with antigen presenting cells, and they will either present something that's caused disease or not. Okay. Um, so the positive selection means that they are capable of choosing something that will cause infection and launching an immune response to it. Negative selection means that they could choose something that is not a foreign particle, that is a self, and decide not to do anything with it. So those cells will lyse. Those cells will stop. They won't proliferate. They'll die. Okay, eliminate themselves, kind of. Which, of course, is very important because if they don't do that, and there are cases where they don't do that, instead they go on, and you get an autoimmune disease, which are really kind of nasty diseases, unfortunately. And also, unfortunately, they don't treat them so well. They kind of treat them just by suppressing the immune system overall in general. It's not really a way of treating. I guess it's going to help and stuff, but wow, people still suffer from those diseases, and you, the long-term effects of treating it that way are pretty bad for the body. Plus, they become immunocompromised. I mean, you're suppressing the whole immune system just to try to get that one response. They become immunocompromised, and it's really a bad situation. So nowadays they're trying to find antibodies to antibodies. Okay, antibodies to antibodies. So the antibodies that the body makes that cause an attack on the self tissue, they treat these people with an antibody to that antibody, and they stop that antibody from binding. And it's really a nice way, but it's very expensive, very difficult, but it's really much nicer, better, gentler, and kinder way to treat the patient um, than just general immunosuppressors. But anyway, hopefully, another thing that will advance, and we can finally be able to treat people who have autoimmune diseases much better, um, and they live better lives. Anyway, um, so again, positive selection, we do choose that one. So what happens is it binds to MHC molecule, yes or no? Yes, that cell survives. Okay, and then that cell is going to, no, sorry, that cell survives. Okay, that cell will survive. That T cell will survive. And when it proliferates, then there are many of them, and it causes a overall response to the immune system. It doesn't bind. Body goes, don't need that cell anymore, so it dies by apoptosis. And then we got negative selection. Oops. Negative selection. And same kind of thing. This is self-antigen. So MHC class 1 molecule. Class 2 dendritic cell presents self-antigen to pre-T lymphocyte. Does it recognize it's the self-antigen? No. Then that one can survive. If it recognizes self-antigen, it's destroyed by apoptosis. You don't want a cell that's going to launch an immune response to react and cause that launch of the immune response to a self-antigen. As it says here, selective loss of either CD4 or CD8 proteins. Most of the cells actually have CD4 and CD8, but whether they differentiate into one or the other depends kind of on the need of the system, of the cells, of the body. And so they'll just simply lose the one that they're not going to be anymore. So then the T lymphocytes migrate from the thymus to secondary lymphatic structures. So they migrate to all of the lymph nodes and the tonsils and all of those lymph nodules that are throughout the body. They end up going into those. And so there they wait until the body encounters that same thing again. And when it does, click, they are ready to just take off. So now they become, if the infection's still going on by that point, they will become like warriors in that. They will kill just those things that are causing the disease, okay? All right, so I'm going to pick this up in part 4.